most respected director of Center for Plant Breeding and Genetics, Dr. Ravi Keshwan, professor and head of Department of Genetics and Plant Breeding and Genetics, Dr. Meenakshi Ganeshan, Dr. Jayamani, and other professors from this division, and also students. And I could see the Dean School of Postgraduate Studies, Dr. Senthil, also online. First of all, I would like to thank thank the director and also the Dean PG Studies for providing this opportunity. Um, uh, in fact, am I audible? Yeah. So you're all masters and PhD students. PhD, all are PhD. Great. So I also did my undergraduate from the same college here, ACNRI Coimbatore. I finished in 1999, 98 December, I would say. And the basic interest in the subject in genetics and plant breeding, I don't know, the present day students would not be remembering, maybe professors may be remembering. Dr. G.A. Palniswamy sir and Dr. Gopalan were our teachers. So that is when we got inspiration and also the idea of the importance of genetics and plant breeding, and some interest in the subject, which we followed it up. I did my master's and PhD at IRI. I come from ICR's Indian Agriculture Research Institute, which is located in New Delhi, and famously known for the Green Revolution. It has been the seat of Green Revolution. Professor M.S. Swaminathan was the nodal scientist who has been influential in bringing the green revolution for the country with the introduction of semi-dwarf photograph of the Imperial Agriculture Research Institute, which was there in Pusa, Bihar. So the institute shifted to New Delhi after the earthquake in 1935, and then since then we are located in the Department of Genetics. We call a Division of Genetics there, and now it has been renamed as Genetics and Plant Breeding. And I did my master's and PhD from this division, and then worked in National Bureau of Plant Genetic Resources, its regional station in Jodhpur, for 5 years, and then once again joined back as senior scientist. And the statue is you see in front of the division was installed last year. So I'll be taking you through the different aspects, and let us see like what we have been able to achieve in our breeding program, and what are the possibilities and what are the challenges in adapting molecular breeding in the regular breeding program. We all heard the name of Charles Darwin, right? So he wrote a book on origin of species, which talks about the origin of variation, and also the importance of natural selection in shaping the variation. So the nature itself has played by and large a major role in shaping up what is to be selected and what is to be not selected. And based on his studies in Galapagos Islands, he proposed several postulates among which the survival of the fittest is the most well-known one, wherein the mutations in general are responsible for accumulation of the genetic diversity in any organism. The ultimate source of variation in any organism is the mutation itself, spontaneously occurring in nature. And this diversity, these mutations are very non-random in nature. It is not specifically happening at, although there are mutation hotspots, but they are not very random in nature. And then the offsprings, or the number of offsprings produced by an individual, is quite large, out of which only certain proportion survives in nature, which he called it as survival of the fittest. The mutant which has the most desirable adaptation for a given condition will survive in nature. So nature itself has been an acting force in improvement of organisms, in animals, in plants, in all living beings. And with the early farmers, with the process of domestication setting in somewhere around 11,000 years ago in the Fertile Crescent, which is now the Middle East region. A large number of relatively small number of wild species of different crops evolved. Here I have given the examples of tomato, which has originated from, now it is Solanum pimpinellifolium into Solanum, and which has given rise to modern day tomato, which is Solanum esculenta. And also Oryza rupifogon, which has given rise to the Asian cultivated rice, Oryza sativa. And this has happened through variation, which has generated naturally. And then at some point, they have been selected by the early hunter gatherers, uh, which provided, like, for example, the Oryza sativa, the cultivated rice in Asian rice, the progenitor of this, there is a species in between this and rupifogon, which is called Oryza nivara, which is the annual form of cultivated wild rices in Asia. So they were selected for a reason that there were comparatively less shattering in nature, which provided the advantage of harvesting in a single go, rather than otherwise you will have to harvest, like in a typical panicle, the flowerings, the anthesis starts from the top to bottom, and it goes on for around 10 days. So obviously for the grains at the tip of the panicle will mature faster, and the grains at the spikelets at the bottom will be filling later on. So that 10 days interval, the top portion of the panicle, if it is shattering, it will shatter. The grains at the top will shatter. The bottom, by the time you wait, harvesting the bottom ones. So it gave a definite advantage in terms of harvesting. You can wait for all the complete plant to mature. This is for only one panicle. And if you look into the differential flowering of the primary panicle, primary tiller, and the secondary tillers, there would be another 3 to 4 days to that. So it will be a period of 10 to 15 days by the time the complete plant matures. And you cannot wait till complete plant matures, because shattering will happen in the early flowered spikelets, which has grains are formed. So this offered an advantage, and so the early farmers, they started selecting it out for the non-shattering types, mostly in. Similarly, there were a suite of traits for which indirectly, based on the phenotype, selection has happened over centuries, which in general are called as domestication syndrome traits, which directly or indirectly is related to establishment of a otherwise wild species into a cultivated crop. So For example, in rice, if you look at the two loci which govern grain shattering, SH4 and QSH1, they were one is in chromosome 1 and the other one is in chromosome 4. And the red pericarp, you would have seen like the traditional land races is some of them they have different colors of pericarp. Brown rice, if you look at, can you name some varieties which are very popular in the market? The so-called healthy rices. Anyone? Any rice which you have heard about, which is supposedly nutritionally enriched and uh, better in quality as compared to the improved varieties, they say. Kauni is one, good. Yeah. Any other? Let us make it a little interactive. Um, Kauni is one, definitely. You would have heard about Mapile Samba, you would have heard about the red pericarps. The pericarps are red, Kauni, it is slightly more towards black. Yeah. There are different shades if you look at the rice diversity. So that pericarp color is determined by RC and RD. And more so in black rice, it is something related to anthocyanin biosynthesis pathway, the chalcone synthase related genes. And then uh, our preference have slowly started different geographies. If you move, you have preferences towards different types of rices. So if you move to the northeastern region, you have preference for sticky rices. Basically, they prefer taking soup, rice in the form of soup. And if you go to northwestern part, the northern India, they like aromatic, long slender, longer cooking rices, which have better kernel elongation. You come down to south, you need medium slender or short slender non-aromatic types for regular consumption. But maybe if you are using it for specialty preparation, then you have short grain aromatic types like Jeeraga Samba. And if you go to Kerala, you have different region have different variability in terms of the amylose content preference, in terms of the pericarp colors, in terms of cooking quality traits also. So GS3 determines the grain length, the GN1A the grain number. These three are essentially the improvement traits which has been mapped and selected later on during the breeding process, but these three has been um, more towards the domestication syndrome traits. Similarly, you have in different crops different traits. For example, fruit size in tomato is one. Okay, and fruit number, fruit firmness, okay, fruit shelf life. All these loci have been part of the improvement exercise during different times. If you look at the different breeding methods available in crop improvement, these are the general methods which we have in the conventional breeding. Uh, the most simplest one is selection. Simply select from the available, the typical pure line selection, which we have all studied, the Johansson's experiment, wherein you do from the available variability in the germplasm or in a given seed lot mixture of genotypes, you try to select the best performing ones based on certain set of characters. Maybe it may be the seed weight or seed number, seed color, whatever it may be the trait. But that selection is purely random. Like you are selecting just based on the phenotype, and it may be heritable or non-heritable also. And this process has been happening even before Mendel proposed his um, theories of inheritance, the Mendelian laws of segregation and independent assortment. So mainly in fruit crops and also some pulses, there are reports. But since the discovery of Mendel's laws, the proposal and rediscovery of Mendel's laws, we have been following hybridization and selection as the most commonly used approach for crop improvement. So you make a cross between two contrasting genotypes and then try to create recombinants through the process of recombination breeding, and then go through pedigree selections, selection in each generation depending upon what the, but primarily there also the selection is primarily based on phenotype. So if if you are going to join a new crop, so I know Surinder here. Surinder, which crop do you work? Black gram. Okay. Surinder is working in black gram. Suppose if he joins as a assistant professor or scientist somewhere in a completely different crop. Okay. It will take, how long you have been working on black gram, Surinder? 2 years. So you have a reasonable idea like how the plant looks, what is the flowering phenology. If you take up sowing, you know the available variability in the germplasm, what are all the traits which are important, all these things you know. Suppose if I ask in rice, what are the important traits? Can you tell? Number of tillers, number of grains per panicle, quality traits. So these are generic. What would be the ideal number of tillers, you think? What would be the ideal pod number in black gram? Around 7 pods per branch. Seeds per pod would be 7. What would be the grain number per panicle? What do you think would be the best number? So the why I'm asking this question is not to embarrass him. The idea is like this exercise, you need experience and also uh, experience for a minimum number of period to get hold of the crop itself. Suppose if you join, rice is still a easy example. Suppose if I ask castor, okay. So when I went to break the monotony, I will take another example. Have you heard the name of mod bean? Any one of you? Mod bean. So you are some of the most advanced in terms of learning, I would say. So I haven't heard mod bean till I finished my PhD. And when I joined the NBPGR, what is the species? Vigna quantifolia. Where are you from? So you know that. That was a no-brainer. So you are also from, you are from here, but still then, well done. So mod bean is a very specific group of Vigna, very specific species of Vigna, which is Vigna quantifolia. I haven't heard the name of mod bean at all till I joined there. And I joined there, and they are discussing, the scientists there, they were discussing like mod bean will take up sowing of mod bean. Mung bean, they talk, copy, they talk, I can relate. But when they said mod bean, for 1 week I was always hearing this term mod. Maybe I thought maybe they're talking in a local language or something, so I'm not able to understand because for us Hindi is also new. We are alien to Hindi also, so I thought maybe some of the crops which we have read possibly their way of pronunciation or interpretation is different. But then when I saw it for the first time there, the crop. So to get an idea of the crop itself takes time. We have all heard about cluster bean, right? Except except that we make vegetable salads out of cluster bean. We are not dealing with cluster bean in terms of day-to-day activities in learning, right? So, but they are very major staple crops of Rajasthan. Like you go there, you have pearl millet, cluster bean, and mod bean. These are the There are certain species which I did not know at all. Like in Citrullus, we are more more conversant with watermelon, right? There is another species in Citrullus which is much more popular in Rajasthan. What is that species? What do you call it? What do you call it in your language? You call it Kalingada, Kalingada, Tumba. Okay, so it is a different species, Citrullus colocynthis. It is very, very drought hardy, and it is a staple diet not only for people but also for fodder, as a fodder. And those seeds are used like we take cashew cakes, right? Cashew burfis. Uh, Many of the sweets which comes adulterated with the seeds of Citrullus colocynthis, because that gives the same kind of consistency when you make cashew burfis. Cashew is very costly. Not everybody can afford. So you use it as an adulterant in making cashew burfis. So the take home message is, in order to practice hybridization and selection in crops, you need, based on phenotype, selection based on phenotype, it takes time and experience to know about the crop, and then to develop expertise on that. Leave aside the transfers happening within the genetics department from one crop to another. It also affects your flow in terms of improvement. So the phenotype based selection, one of the major bottlenecks is you need certain period of experience in order to exercise selection, and also to identify parents which can be used in crossing. That is the take home message. Okay, and wide hybridization, it is still one more step of difficulty in terms of compatibility between the gene pools. Okay, and also if it is a cross with a wild species, then you will have to look into the so-called traits which have been avoided during the domestication process, like shattering, or if you take cucumber, modern day cucumber, right? Cucumber is sativas. It is very sweet and juicy to take, right? But the progenitor of that is Cucumis hardwickeyi, which is like shorter fruits. It is more elliptical, and it is very bitter in taste. Sorry. So you have to, when you make wide hybridization or crosses involving different species, you will have to take into consideration the compatibility while making crosses, and also the linkage drag when we are transferring any traits. And hybrid breeding, provided if we have a mechanism or a method to control the pollination, you still have to make crosses, random test crosses, in order to identify combinations which can make good, which can yield better. So it takes time to, it is very difficult even now to predict which combination will work, as which hybrid combination will give you the best hybrid. So we have been dealing with random crosses all the time in hybrid breeding, except except some of the more advanced programs in private seed companies, where they have kept the parental pools apart, and so they know that there is diversity between the pools. So one parent from each pool made a cross, you get heterotic combination. But there also the extent of heterosis varies, and till now we don't have a handle. In conventional hybrid breeding, if anyone with whatsoever the kind of experience they have, they will not be able to tell this is the combination which is going to give me the best hybrid in terms of yield, and this is the parent which I'm going to choose. And another approach which has been very popularly used has been mutation breeding, which is for crops where you have hybridization is very difficult in crops like finger millet, or in the minor millets where the variability is less. You depend upon mutation breeding to create the variability itself. So there also the mutagenic agents. What are all the different mutagenic agents which are used? Any idea? Can any one of you help? Mutagenic agents used commonly. EMS is the most commonly used one in chemicals. In physical mutagens, gamma rays are the most widely used. But can you can you with surety tell, like, if I create, you have a LD50 dose, right? You have an LD50. What is that? Lethal dose. So 50% of the population gets killed. There is a variability between genotypes for this LD50 value also, right? And and then there is no surety that you are going to create the mutation which you want. You are just randomly trying to hit something and hope that you are getting the best, right? And also the number of mutations which are useful can be very, very less. Most of them, since we are, the basic principle of using mutants itself is based on lethality. Almost 50% of the mutants are already gone because it is already killed during the process of selection itself. So this is the limitation of mutation breeding. And polyploidy is another level of difficulty. The first man-made synthetic polyploid, reticale. Yeah, it took you have an idea like how many decades it took for generating triticale itself. There has been similar attempts by crossing potato and tomato to get tomato. Reverse happen. Okay, they didn't get a tomato fruit. They got in the initial cases they got potato flowers and tomato roots. Not useful. Okay, so it needs very massive efforts, and there also you are not sure whether you are going to get the product which you are going to which you aimed at. Okay, so if you look at the general limitations, is selection is primarily based on the phenotype. Gene transfers are limited by sexual compatibility and linkage drag, and the mutations are random. And in general, they are low in frequency if it is spontaneous mutation, and then the usefulness is questionable. And then with conventional breeding process, it takes time. Okay, first leave aside the time taken by one to get an experience on the crop itself. Even if you are an experienced breeder, um, our teachers and researchers will tell you like it takes 10 to 12 years in the conventional breeding to come out with a product. So in breeding, the basic thumb rule is the variety which you are releasing as you as a first author or you as a lead breeder is not yours. It is somebody else's work which we are trying to identify and release. The work which you are going to do will be getting reflected 10 years down the line through somebody else. So it is very, very important to, but the basic aspects still remain the same. Okay, I will just take through the basmati rice improvement. Having said that, having said that, it is not all bottlenecks everywhere. There has been very, very significant improvements made in through conventional breeding, even though it has taken time. Okay, I'll take through the story of basmati rice, the program which I have been associated with as a student since 2001, and then later as scientist. This is the photograph of a traditional basmati variety at maturity, not even maturity, at grain filling stage. So they are all tall. So a slightest wind or rain, it lodges. Even at flowering stage, it can lodge. It is a field full of crop, but flat lying on the field. So because of the height and the weight of the panicle, it lodges. Stem also are weaker. They are photosensitive. It takes around 160 days, the traditional types. Here also we have traditional varieties which take photosensitive type. And then yield levels are somewhere between 1.5 to 2 tons, which is low comparing today's standard, right? And basmati is uh, product which is protected under the geographical indication in the 7 states. In Jammu and Kashmir, you have 3 districts: Jammu, Katwa, and then Himachal Pradesh, Punjab, Haryana, Delhi, and only 30 districts of Western Uttar Pradesh and Uttarakhand. These are the regions earmarked for GI of basmati. Now the question for you: can anyone tell one GI product from Tamil Nadu? Hmm. Agricultural product which has been granted GI. I want to break the monotony so that nobody is sleeping. Any GI product, you are all PhD students, right? You did IPR course, right? Yeah. Any GI product. Any other volunteers? Where are you from? Where are you from? Yeah. Coimbatore. Okay, you are from Coimbatore. So Coimbatore, what product is there? No, sorry, Coimbatore is not a good choice. Anyone from Madurai? Yeah. Any GI product from Madurai? Jasmine. Anyone from Tiruvarur or Nagapattinam or Mailaduturai, somewhere near that area? No. Okay, we have a lot of products which have been granted GI. In paddy, we have Zira Samba, which has been granted GI. Kauni, possibly there are attempts to make. Jasmine is one. Mm, Nilgiri tea is. Erode turmeric is another one. Yeah. Uh, I think Sirumalai banana is also earmarked with GI. What it gives is GI products give the authenticity for the product, that GI mark, and also premium price for the product. What happens in agriculture? If some product is produced in large scale, automatically the price comes down very, very drastically, right? You have seen recent price crash in onion. Onion, I suppose it is less than, you would have seen in all these WhatsApp university that some farmer sold some 2 quintals or 2 tons of onion and he got 58 rupees check. No, it was very widely in the newspaper, right? So if the production is higher, if value addition is not there, the product generally the price goes down. That is how economics work out. But with GI, what you have is you have area earmarked, and the produce from that is only commercially labeled and sold as that product. So that gives certain extra value to the product itself that way. Okay. So this is the GI earmark for basmati, and the basmati breeding program started sometime in 1966 uh, uh, with Professor Swaminathan as the program lead, and we had Dr. E.S. Siddiq. Dr. Jawahar Ali is here. His father was one of the team members in the Basmati Rice Improvement Program later on. He led the program after Professor M.S. Swaminathan. So in general, the notion was that you cannot combine quality with quantity. Okay, if it is a quality product, you cannot increase the productivity beyond certain level. That was the notion during those times. That was Green Revolution era. TN1 was one of the uh, first products released in the country as a semi-dwarf variety. Taichung native one. The SD1 gene from DG was transferred, and TN1 was created, and it was imported to India. So that was used as a dwarfing source, and then there were several studies which large number of students. First, Dr. Siddiq worked with Professor M. S. Swaminathan to look into the basic traits which define the basmati quality, followed by a suite of students who looked at the inheritance of different traits, the aroma, the grain length, the kernel elongation, the amylose content, the alkali spreading value, all these traits. And then it took almost 25 years to come up with the first product, first semi-dwarf basmati rice variety. So when I said hybridization and selection, it takes time. Leave aside the basic genetics worked out. If you have some idea, somebody else has done that work, you have some handle. Otherwise, you're completely in dark. You have to do everything yourself, right from the working out of the genetics of each of these traits. And this was the first semi-dwarf variety. And I have to rush it up, I suppose. This is Pusa Basmati 1121, which was released in the year 2000, 2003. This is one of the highest sold variety and exported variety. I will tell you what. Whatever biryanis you are having in any shop across India, across the world, it is from this variety alone, and a couple of other varieties. And this is another variety which is a short duration variety, which is Pusa Basmati 1509, which is 120 days duration as compared to 160 days in traditional varieties, right? And this is the improvement which has happened in kernel length and also cooking quality after cooking, like the grain kernel length after cooking, the cooked rice kernel length. So this is the traditional one, Basmati 370. PB1 was released in 1989, and then 2003 PB1121 was released, and it was a game changer for some reason. And the major reason was the cooked rice kernel. You cook one cup of rice, you get 4 cups of cooked rice. And in the hotel industry, the better the volume, the higher the value of the product, right? And everywhere, anywhere you go, the biryani is from this variety, PB1121. And then this is PB1509, where we have the same grain quality, but duration is reduced to 120. Here it is 145 days. It is tall and largest. It is semi-dwarf and comparatively less in lodging. And this is the figure showing the yield level of Tarori Basmati, which is around 2 tons, 2.5 tons in 160 days duration. PB1, the duration was reduced to 140 days, and the yield levels had gone up to around 4.5 tons. And with 1509, the duration came down to 120 days, and the yield levels went up to 6 tons. These are the mean yield levels. And then if you look at the net return per hectare per day, it has increased from 688 to 2,250 rupees. So this has happened due to value addition in terms of the grain cooking quality, and also the concomitant development in the rice industry also. And when there is a good product, then everybody wants to take that. So this, when PB1 was released, we were exporting somewhere around 3,553 100 tons, around 35,000 tons, with 317 crore rupees. So any variety, when it is released, it takes 4 to 5 years to get into the market, they spread, and then to create an impact. So the PB1, with PB1, the volume and value increased almost 2.5 times to 865 crores. And with the release of PB1121 in 2003, you can see the jump in terms of volume and value. We had last year we had around 4.7 million tons of basmati rice exported with a value of around 34,000 crores. And this has happened with conventional breeding alone. Uh, they Professor MS started the work. Dr. Siddiq, till he became the director of IIRR or DRR, he was there. And along with Dr. Siddiq, Dr. VP Singh was there since 1965 till 2007. All his lifetime, 35 years, he had been working in rice and only on basmati rice. So with that kind of experience, you tend to accumulate knowledge and expertise with which you can move forward. But if you start moving from one crop to another or one institute to another, that kind of improvement cannot happen in conventional breeding. Okay. Now with any crop, there are different diseases. In rice, there are blight, blast, sheath blight, false smut, bakani, bacterial grain rot. And this is one disease which we had studied in only in plant pathology courses. I have never heard about this disease after that in undergraduate plant pathology courses. Foolish shielding disease, right? What is foolish shielding disease? It, it is Bakane disease, right? So what happens is plant starts accumulating more GA. So the plant grows taller and then it exhausts all resources and dries off. So annually, this was the status when sometime in around 1999, annually around 15,000 crores have been utilized for management of diseases and pests in basmati rice. Basmati rice alone. Leave aside other rice crop across India. But fortunately, we have a large number of genes which have been mapped, and also QTLs for mainly for blight blast. And blast is such a disease that when it this is blight, when it occurs, the leaf dries and completely the crop is completely dried off. If someone has the farmers ultimately have to set it on fire. And if it occurs on seedling stage, it crosses crescent, the seedling won't grow at all. Okay. But for, and the PB1, which was released in 1989. So when a crop is good, when a variety is good, it starts growing in large area. And then automatically the weakness of the variety comes to get known. Uh, so it was highly susceptible to bacterial blight. And this is the field of PB1 sometime in 2000 also. And fortunately, we had, at that time, we had around up to XA21 were available in the differential set, which was developed by ERI. Okay. Now we have around 42 genes, R genes identified for bacterial blight, but there is no source in basmati rice. The initial work was carried out with the differential set to identify what are the genes which are effective, showing resistance against the most virulent isolate collected from different parts of basmati growing regions. And they found out XA4, XA13, and XA21 being resistant. And the interesting feature is that XA13 and XA21 was able to provide a resistance reaction which was similar to all the four genes together, XA4, 5, 13, and 21. So if you are in a position to decide which one to do, whether you will take two genes or four genes for incorporation into PB1. Good. That is one good explanation. There is no right or wrong answer. Let me tell you. You have to justify why you are doing that. That is the only thing. Any other answers at all? What is your good name? Tamil. Tamil. Okay. Tamil. Suppose you are posted in TRRI. I'm taking a scenario. Okay. You are posted in TRRI and you are given all these three. And ADT 58 is already released, I suppose. So it is susceptible to bacterial blight. So you will take 4 genes, the IRBB60 and make crosses, right? That is what you will do. So F1, what you will get? Heterozygosity for all. Here there is a mistake. XA4 is a dominant gene, so it should be capital X. Uh, in F1, what would be the genotype for all the 4 genes? They will all be heterozygotes, right? So you make another backcross because you have to improve ADT58. So what you will get in BC1F1? What would be the segregation ratio? Like for there are 4 genes segregating, right? So one parent is homozygous recessive or homozygous alternate allele. For a dominant gene, it has a recessive allele homozygote. Other parent is hetero. So you'll get 1:1:1:1. That would be the proportion. So if you have 100, you will have one, which will have all the 4 hetero, right? And the other extreme will be all homozygous recessive, right? So the proportion of individuals will be comparatively less, which you will be able to select as heterozygote for next back crossing, right? So that is one. Okay, leave aside crossing itself. Okay, and then suppose you are taking to BC to F2, you have markers. You suppose if you take that scenario, you are using markers also. So you have 1,000 population of 1,000, you will get around 250, which are hetero for all 4. So what you will do? You will make another back cross. Okay, your probation period is completed. You are not happy with Aduturai, and you apply for transfer and go somewhere. Coimbatore, there is some posting. You come here. What will happen? Okay, it is possible. It is possible. It is there. Okay, but uh, permitting more genes provides value. In long term, it has sustainability. It can sustain evolution of new races. But the first objective should be to control the disease itself. So you have to choose the easiest option first, and then if these 2 genes succumb, then you have a backup option of XA5 also available, XA4 also available. If you in the first case itself you expose all the genes available for a given disease, and if there is virulence evolved for all the 4 together, then you have nothing. You will have to move to some other genes. You have options, but still it would be good to do with 2 first and then keep stacking on top of that. Okay, that is what was planned. And a typical marker arrested backcross breeding. In a conventional backcross breeding, you go up to which backcross to reconstitute, you go up to 6, right? But markers offer some advantage in terms of selecting for the target loci as well as the background, and also for recombinants. Okay, these are the 3 different selection steps during the marker arrested backcross breeding. So foreground selection is for the target loci. You select using a marker which is gene based or gene linked. Background selection is the one which you do it to recover the recurrent parent genome across the genome, and then the recombinant selection to avoid any linkage drag if it is any. Okay, yeah. So with that, even you can do that within 8 seasons. And if it is a crop of 120 days duration, in Aduthurai, you can take 3 crops. Here in Coimbatore, 3 crops per year. So within 2.5 years, you will be able to come with a replacement. So it is also how fast you develop a variety and how efficiently you try to replace the variety, which is important in making these decisions. This was the strategy which was available. You do marker assisted foreground, background, then phenotype for the disease, and then agronomic traits, quality evaluation, and then release. Then during the process, what we found was there is one critical step which is missing, which can offset the background selection or complement background selection to a great extent, and that was phenotypic selection in the backcross one stage for the plant type, for the grain type, which you can do in the BC1 F1 stage. Thereby, you can reduce the number of backcrosses which needs to be made. That is one important step which we did in the backcross breeding itself. And then this paper came in 2008, wherein we introduced two genes, 13 and 21, into PB1, and this variety was released in 2008. And on your this is IR24, which is the susceptible parent, and IRBB 13 and 21, and I think the second last one is improved PB1, and the PB1 is at the last on the right hand side. So this was released in 2008. Nobody had any idea about marker assisted breeding when it was done. And one of my senior PhD student initiated this study, and I was fortunate to carry it forward during my PhD study. And then with the release of improved PB1, there were no guidelines in the system of testing for, the system of testing, trialing in all the coordinator research projects, right? Typically, you have an initial varietal trial, advanced varietal trial 1, and advanced varietal trial 2. 3 years of testing. If it is good, better than the popular checks, then it gets released. But these are different products, right? These are marker-assisted mass-derived products, and then they have been improved for a particular trait of an already popular variety. So new guidelines was evolved for testing these in the ECRIP system. This was set up in rice initially, and now we have in many crops. We have in maize, we have in wheat, we have in pearl millet. Many of the crops, even in chickpea, also they are adapted. Initially, we used AFLP markers, the amplified fragment length polymorphism markers, which are random for background selection. In the next study, for improving the parental lines, we used SSRs, because those markers, by the time we had access, we had projects, money available, funding available, better markers, so we started using that. And there are large number of genes. You have to be very wise in choosing which gene to select. And among the bacterial blight resistant genes, XA13 and 38 and 5 are the most highly performing ones. XA21 and 33 are still okay. In blast, we have PI9 and PI2 and other set of genes. And this is a variety BB25204, which is this photo. Is on your bottom corner is the is the blight infected crop. On the top corner is the blast infected crop, which is very rare to see. And this photo was shared by Dr. Robin. He shared it during one of our interactions earlier. And yes, we had a project of creating near isogenic lines wherein we transferred 7 different blast resistant genes in PB1. And all these different genes are there in the basmati background now. So this is the near isogenic lines in the field, and if we remove these lines and also possibly the labels, you will not be able to differentiate between the recurrent parent and the near isogenic line. This is the kind of uniformity you are looking in terms of morphology as well as in terms of grain quality. But sometimes you have to come across situation like this where the donors doesn't have the grain quality which you prefer. These are all red pericarp types. These are lines monogenic differentials which have the genes for PI9 and PI5. Red pericarp, short bold grains, very poor in cooking compared to PB1. You can look at the cooking. So selection has to happen parallelly along with the selection for the gene in the backcross breeding. And this is PB1 in the Uniform Blast Nursery in Malan, which is a hotspot in Himachal Pradesh. And this is the improved nil with PI9 gene at the same location during the same season. And we also confirmed it through artificial inoculation. I will rush through it. Nowadays, there is more emphasis towards direct seeded rice because transplanted rice is thought as a culprit for a lot of water usage, and there is limitation in labor, availability of labor for transplanting. All these things are moving us towards direct seeded rice. And one of the major culprits or major bottlenecks in direct seeded rice is weeds. And in one of the projects in DBT network project, Dr. Robin here and Dr. R.P. Sharma here. Dr. Robin was the head of the paddy breeding station earlier when they identified a mutant which in the Nagina 22 which can tolerate imidazopyr spray. And this mutant has been named as Robin in his memory. And this genetic stock was utilized for transferring the tolerance. This is Robin and this is PB1121 into 1121. This is with the spray of imidazopyr spray. And a typical marker-arrested backcross breeding was adapted. And oops, sorry, there should have been a video. There was a time-lapse video which shows the difference between the unsprayed PB1509 and the 1509 and the PB1985 it has the gene for blast resistance. I don't know, for some reason it is not opening. So what happens with imidazopyr spray? Rice is normal rice is susceptible to imidazopyr. So this herbicide binds to acetonolactate synthase, one of the enzymes, which is in the pathway of producing leucine, isoleucine, and valine, which is essential amino acid for plant growth. So when you spray this herbicide, it kills the plant. So when you don't spray, it is normal. And when you introgress the gene for this, the mutant allele of AHAS into 1509, even after spray, it remains normal. It is as normal as the unsprayed variety. And there are two varieties which have been released, PB1985 and PB1979. And this is under direct seeded rice. This is transplanted and sprayed. This is unweeded control, and this is 1509 sprayed with imidazopyr. These are the improved versions, tolerant ones, which are tolerating. So far, we have released 11 basmati varieties by transferring different traits into different genetic backgrounds through marker-arrested backcross breeding, right from bacterial blight to blast to both together. These three varieties were released in 2022, and this has been released again in 2022, and this is just released. This has a QTL QDTY 1.1 for reproductive stage drought tolerance. I don't know, maybe this video may work. Yeah. And I said three varieties with both bacterial blight and blast resistance. And we had the farmers Mela on March 2, 3, and 4. There were around 1,000 farmers wanting to take the seeds of these varieties, but because the seed quantity was less during the first few years, the seed quantity is less. We sold around 2,000 quintals of maybe 1847, but even then there was, sorry, there was, till the last day there was crowd. Uh, we have done this in BPT 5204 also, 3 genes for blast and 5 genes for both blight and blast, and in 2 Sugandh varieties also. So the process itself, in the pedigree breeding, on an average, it takes 10 to 11 years, including the testing process, to release a variety. If a variety is already available and we just need to improve for a set of particular traits, then we can still do it in 6 years time. But with the tools which are coming up, like genomic selection and double haploidy, this will go down to 6 years even for developing new varieties through the process of genomic selection itself, wherein you create large number of lines through rapid generation advancement or DH, and then select based on the genotyping and the genotypic predictions, and then release varieties. After release, it is important to track the presence of genes in the varieties during the maintenance breeding cycles. And that is done through quality evaluation and the checking for the presence of genes in the nucleus seed lot as well as randomly in the breeder seed lot. I will stop here for want of time. There are so many other slides, but yeah. If there are any questions, then we can get on with it. Before that, I would like to move to the end. Yeah. This is our team. We have Dr. A.K. Singh, who is the team leader and director now of Indian Agricultural Research Institute. And we are a team of 5 scientists, and all others are students or SRFs in the projects and these skilled helpers in the project. And we have fantastic collaborations. It is very important. No one can have gone on the era where Sanger went on to win Nobel Prize on a single intelligence of a single fellow. Nowadays, the field is expanding so fast and so diverse, and also you need competence at different levels to take the program to a next level. So we have fantastic collaborators within the institute. We have we are totally 6, including me till here. Dr. Nagarajan is in IRA regional station Aduturai, and Dr. Seth is the maintenance breeding seed production in charge in Karnal. Dr. N.K. Singh, Dr. Mahapatra, T.R. Sharma, they are all assisting us in the molecular aspects. Dr. U.D. Singh, Mandal, and Vishnu and Prakash are in plant pathology. Dr. Madan Pal and Vishwanathan are in plant physiology. Dr. Subhash Chandra in entomology, and then Dr. Dinesh in agronomy. And besides that, there are certain locations which are very, very advantageous in terms of screening. Palampur is one location for blast. We have Godalur in Tamil Nadu, which is very good. So there is our collaborator, Dr. Atur, there. From TNA, we have been having fantastic collaboration since the time of Dr. Robin, Dr. Ravindran, and Dr. Manonmani. We interact, and we have projects which are interinstitutional projects. We keep interacting with each other. Dr. Mandal from Hazaribagh and Dr. Padmini from NRRI Cuttack, and the whole group. And thank you very much. And if you have any questions or clarifications, I'll take it. I'm sorry I got delayed by half an hour. Yeah, but hope it was useful. If there are any specific questions or clarification, you have to ask questions. Otherwise, the Viva will not proceed. Yeah. You can ask, you have to ask questions, otherwise you are not going to progress. Okay. So it is not that whatever has been told is correct or whatever has been said is imbibed. Okay, there will always be doubts. So always ask questions, even if it is a, even you think it is a question which is not good. There is no good question or bad question. Question is a question because we don't know. So always have that habit of asking questions, which is good.
Dr. Gopal, can you hear me?
Yes, ma'am.
Dr. Gopal, can you hear me? This is Dandapani from Madurai.
See, 1,000. Yes, ma'am. Yeah. Grain quality, no, we didn't have any issues because we have been purposefully selecting for grain quality. That has been the emphasis of the program itself. So we did not miss there, but yes, we missed. There are a couple of cases. I had slides for that. For a want of time, I had to skip that. It is not always that a new methodology comes in and everything works perfectly well. It is never the case. Improved PB1, variety was good, quality wise it was good, yield was good. Farmers were very happy one season. Next season, first season it went to the field, next season they came back and they said, some group of farmers, they said we are not growing in western part of Uttar Pradesh. Asked the reason why. You can imagine why. We were doing selection on single plant basis. Okay, when basmati is kind of free thrashing in nature, what they do is they harvest manually and they make piles of heaps and then they beat manually. And in general, basmati, if you beat once, all the grains are in the from the panicle is in the floor, thrashing floor. But this variety, it had to be beaten twice. So the laborers, they demanded twice the price for thrashing. And in the breeding process, we never know because we have been hand thrashing labors. So that is like take a panicle and thrash it like this, not through beating. We never realized it at all. But when it went to the farmer's field, this was the feedback. And some of the farmers are engineers in the sense they look at other means. They some area it got adapted well. Then we went and asked why some farmers are happy and some farmers are not happy. Western UP, no mechanical harvesting available. You go to Haryana and parts of Punjab, mechanical harvesting. The price difference between per quintal between the manually thrashed and machine thrashed combined harvested paddy of basmati is between 100 and 200 rupees. And those farmers, they said we are getting better yield and we are able to clear the field faster than manual harvesting. So they do, even if we get 100, 200 less, no problem. We will do machine harvesting, machine combined thrashing and vacate the field for wheat crop. But these farmers, they didn't have access to that. They said, no, no, we are going by manual thrashing only. So they were able to find solution for that themselves, but some of them they could not do. And then, as I said, experience in the program. Then on 1121 is much more easier thrashing than PB1 because PB1 you would have seen it has ons. So ons in general is for thrashing also it is not good quality. 1121 is onless and it is very free thrashing. Even if you leave it in the field after maturity for a day or two, it will fall down, grains will fall down. That was more complex, but we got the lesson in that one and applied in the next one. So the next product we were very conscious in the station trial stage. We were asking laborers to thrash it and see whether it is equally good in terms of thrashability also. That is one case. The other case is Case of Swarna sub 1, submergence tolerance at seedling stage. Swarna, the name itself, the actual name of Swarna is something else. Anybody else can tell? Swarna is a common name, popular name. Okay, it is MTU 7029. Okay, it is from Maruthera station, and 7029 is the pedigree number. And Swarna, the name came because it was very high yielding in nature, and the husk color is golden brown in nature. Golden husk. The paddy husk color is golden in color. So it was yielding good. It was golden in color, and it was also giving better profitability to the farmers. So they named it Swarna. Locally, the farmers named it as Swarna. Still, it is in Gazette notification, it is called as MTU 7029. They write in brackets as Swarna. And it is known for its husk color. Sub 1, the sub 1 loci, the allele which governed tolerance to submergence, it was tightly linked to the hull color loci. So when they transferred sub 1, they transferred the hull color of the donor, which is chalky white in nature. So when it went to the market, the traders, they see the paddy and price it, right? They said, you have brought something else. We cannot give you the price of Swarna. So that was another case of linkage drag into. And another one example is when we did conventional breeding, semi-dwarfing gene SD1 was introduced from TN1. Digivision initiated from TN1, right? So we started invariably using SD1 across all the varieties which have been released till date, right? In the process, what has happened is SD1 is very tightly linked to the QTL QDTY 1.1, which governs the tolerance to reproductive stage drought stress. We brought in the susceptible allele of that loci in all the varieties which has released since then using SD1 as a source. This was identified very recently in 2010, and then we had to break down the linkage between SD1 and the QDTY 1.1. It can happen without knowledge. It can happen sometimes. Even PB1121 is a variety which is, you had seen the cooking and the grain length, right? It yields okay, and the breeder of that variety, Dr. VP Singh, who has been awarded with Padma Shri for developing that variety alone, okay, by the government of India. He says, in his opinion, PB1121 is a choice which is not the ideal. He says it is a give and take kind of choice, because that is giving better profitability to the farmers and all the stakeholders involved. But it is not the ideal case scenario. It has so many drawbacks in terms of free thrashing, in terms of height, it lodges, in terms of susceptibility to disease. PB 1121 is variety which, like, literally everyone started growing in the Basmati region, and because of monoculture of that variety, the disease which I said, Bakane disease, it came into prominence because of monoculture of PB 1121. And now we test all the basmati varieties. All basmati varieties are susceptible to Bakane disease, and we have to bring in resistance from somewhere else, which we are doing. So you may not get the ideal choice at any point of time, be it a variety or your life partner or your guide or your course or your whatever you want to do in life. You have to go with the best available option at that time to go with, and that is life. Okay. Yes, ma'am. That I heard that, but yeah, the available among the available options, we have to go for the best options. That is the one. But not only that, sometimes what happens, uh, the mindset of the farmers is in such a way that they have looked at that variety for like 20-30 years continuously. So they can just grab the one, if there is one change or in any of the traits at any stage, they will tell you it is not good. Just when it goes to farmers field, you will get the real feedback. No, ma'am, you have to weigh in your options, and you have to look at the progenies available at BC2 F1 stage. Whether you have got sufficient recovery in the genome background, the plant type. No, you will not get a isogenicity in two back crosses. That is impossible. You will still have some donor segments, but is that affecting your final product is the question. That is, if it is affecting, then you have to definitely, in case of herbicide tolerance, we have gone up to BC4. BC4 F5 was released as product. So it depends upon, because the source from which it was drawn was Nagina 22, which is a non-basmati, and many times there are. That is what I say. Like, if you look at the scenario and then plan accordingly. BC2, if your donor is also elite, you can uh, uh, still be happy with that, because if the source from which you are drawing the genes are elite as well. So now we have a pool of basmati varieties with blight, blast, now for drought also. At this stage, if we have to make backcross, we may not go to BC2 also, BC1. Or better, you go for forward breeding. You make a cross and get F2 and then select the basmati grain quality, and then select for the target genes, and then get into a new product itself rather than going. Marker arrested backcross breeding is just a compromise to till the point the next best variety comes through a pedigree breeding. So it is not a complete solution itself. It is just a solution for a short term period. But we need to have a pool of elite lines possessing the traits and the favorable alleles for all these diseases, pests, which are very common. Till that point, you don't have option, because for me, if you ask me, I will be very happy. I will say BC6. Yes, we have to do BC6. But for BC6, it takes another 3-4 years for a product to come out. By the time your crop is gone, that variety is vanishing from the picture itself. You don't have any replacement at all. So you have to look at those kind of scenarios and then decide. Like we had this southern China black streak dwarfing virus disease, right? In parts of northern India, we had in non-basmati varieties. The plant grows this tall and then it doesn't produce any yield at all. This is a new disease which has been diagnosed only this year. First of all, we could not diagnose what disease it is, whether it is a physiological disorder or anything else. Then this disease has been identified last year, and now we have to make a decision which one to grow. So our option available is the next best possible option. Which of the varieties are comparatively less susceptible? Take that. Now the basic question itself is, we are estimating recovery. If you start asking that question, the question will be to the next level. The number of markers used for recurrent parent genome estimation itself is a bias, right? We are not covering, unless we are going for like whole genome resequencing kind of situation where you look at the whole genome itself. You are only looking at, if you are using SSR markers typically. In the first case, we got around 62 SSR markers from around 600 SSRs available at that time in 2001. That was the highest possible at that time. And then improved Samba Masuri, which was also released through markers, but they had only 35 markers polymorphic, because at that time the resources available and the constraints with which you are working determines that. But now you have 50K SNP array, you have 80K SNP array, but do you have the money to work with that kind of arrays? That array is a single array of 96 samples to be analyzed, cost you somewhere between 6 to 7 lakh rupees. So if you have to do one in backcross one background selection using the 80K array, then what you'll have to spend is for 96 genotypes, you have to spend 7 lakhs. Whether you have that kind of money to assess? So it is just looking at the available options and what is best possible we select. If you ask me, ideal case scenario would be completely different. Why to use markers at all? You resequence the progenies and then look at the genomes and then see. But the larger question is whether you are breeding for the sake of markers itself or you are breeding for the product. Which would be the answer? Which would be the answer? Product. So that is the answer. So if you are happy with the product, the stakeholders who are growing the product is happy, then nothing like that. I will tell you the case. 1509, PB 1509, 120 days duration, very good yield, very highly economical, and it escapes blast disease because it is harvested early. Okay, but what happens when we introduced these two genes, four genes for blight and blast, two each for blight and blast, we had several progenies, 120 days also, 126 days also. There was a delay of around 6-7 days, but this was found to be the highest yield and better tolerance to disease, and grain quality was also very good. So we said we will take a chance, because ideally speaking, we should have gone with a genotype which is 120 days same duration, but we had 6 days extra. But still we looked at it. It gave some other advantage in terms of yield. So we said it is a compromise. So let us try it. We had several nils, but we tried. What happened was that yielded among all the varieties last year, that yielded the highest, and the demand for that this year was like, already we have sold more than around 4,000 quintals of seeds to farmers. So sometimes you have to take decisions which are not strictly through the process of looking at isogenicity or near isogenicity in question, but it can be based on the product which is there. Yeah, if it is advantageous, you still can take traits from the donor also. Yeah. So GA is basically granted as a protection mechanism for the traditional growers, in order to acknowledge that they have been responsible for maintaining the practice of growing or producing certain product right from their time immemorial. Like, it is associated with the heritage of a particular region. And for basmati, it is the weather, it is the soil, it is the water which is responsible for such a good quality grain. And more than that, it is the heritage associated with that region. It is a protection mechanism to acknowledge the traditional farmers, that they have been protecting those cultivation of those varieties since time immemorial. It is just acknowledgement of that, and that is the reason for it. You can still grow it, but you cannot sell it as basmati. Commercially, you cannot sell it. There is lots and lots of demand. Like, farmers from everywhere, they want basmati. I have seen from Kashmir to Kanyakumari, farmers coming from east to west. Everybody want basmati. Not only from India, from abroad also they want to cultivate basmati one. But it has, to be frank, um, if an industry or a product has to grow, around that product everything has to grow. It is like you are looking at an iPhone and then you are looking at some other phone, some other brand phone which is having similar features but costing is very low. The whole ecosystem determines that, right? So the basmati, it is not the variety alone. The different stakeholders who are associated, right, from millers to exporters, the quality control mechanism which has been established in that geography is very, very, at every stage there is a checks and balance to eliminate lower quality, lower grade rices from it. It is not that entire region cultivates basmati. There are non-basmati rices also cultivated in that. But there is checks and balances already available to maintain that product quality. And so, if you ask me, any region within India has their own specialty rices, and market will develop once you have products in hand and start working towards it. Now I heard that for Jiraga Samba we have Vaigai Dam 1 as a replacement. So once you start bringing out those kind of products and it gets acceptability at different stakeholder level, then it will gain popularity and and an industry will develop around that, and then it creates that market value for a product. We have Kauni, we have BPT 5204, which is a huge segment, medium slender grain segment, where everybody wants that. It is not that because of that only. It is associated with all levels of, right from producer to consumer, all levels. So that kind of development will happen only when all are involved in the process, and then it goes. And there are specialty rices across India which can gain the same traction. Now I don't know, like at least 100 farmers they came asking for black rice, black rice in the Mela. They say black rice, they say 150 rupees, 300 rupees. You have to always look at from this angle. If you produce less, you will get better price. If you produce more, automatically the price, that is the economic, that is the way economy works. But then if the growth is in a steady manner, then the market also develops and builds up for such kind of. We had only hardly around 35,000 tons in 1987 80. Now we have 4.7 million tons. It has not gone from there to here within 1 or 2 years. It has taken like 40, 50 years. So when product starts coming, it will automatically get the market value also. So the Manipur black rice is a GI product of Manipur and uh, some parts of Nagaland. And you cannot eat Manipur black rice all the time. You cannot eat Kauni for all the times, because anthocyanin itself has a bitter taste. You have grapes, black grapes, you eat, you will have that bitterness. Same bitterness is there in black rice also. But it is used for making sweet dishes. So you add sweet, it offsets the bitterness to a level. So you use it for making payasam or kheer or whatever it is. But you cannot eat payasam all the time. Only payasam only I will consume. It is not going to happen, right? It has to go within the Indian food system which we are using. For a variety to perform here and to gain traction in the market, it has to go well with sambar, rasam, and then curd also. It cannot go with only sweet dish. It cannot happen. Maybe people will project it in a different way and try to push, but that push can help only to go to a certain level, but it cannot go beyond a certain level, because automatically people will realize, right? You cannot eat anything which is, because our digestive system itself is it has got adapted to white rice, milled rices. Now going back, you have to change the complete psychological profile and also the gastric profile of the complete consumers, which is not going to happen overnight. Today morning I had, for that matter, today morning I had a idli made out of mole sambar, which was looking red and white. I said, what is this? So they said, oh, this is from this. But it will take time, yeah. Thank you. Thank you very much.
So on behalf of the Department of Genetics and Plant Breeding and Directorate of CPPG, and on behalf of the Dean SPGS and the University Professor, I thank Dr. Gopala for giving valuable information on what is happening at JRI at the global level. So I thank once again for the informative and very good lecture.